What does it take to challenge the deadliest brain tumors with sound waves? How do you scale a MedTech startup into a multinational clinical company? And what advice can a top neuro-oncology innovator offer to those hoping to follow a similar path? Welcome to Neurocareers Doing the Impossible, the podcast where we explore the extraordinary journeys of those advancing the future of neuroscience and neurotechnology. I'm your host, Dr. Milena Korostenskaja, and today we are speaking with Dr. Michael Canney, Chief Scientific Officer at Carthera, a clinical stage MedTech company revolutionizing brain therapy through therapeutic ultrasound. With over 15 years of experience in MedTech innovation, Dr. Canney specializes in glioblastoma, neurodegenerative diseases, and therapeutic ultrasound drug delivery. He is currently leading the SONOBIRD Phase 3 clinical trial across 40 sites in the US and Europe, pushing the boundaries of what's possible in treating recurrent glioblastoma. Michael's career spans everything from FDA regulatory strategy to high-impact academic collaborations, and he has co-authored over 50 peer-reviewed papers in top-tier journals, along with 14 patent families. But what truly drives him is improving patient outcomes and bringing hope to some of the most challenging areas in brain health. Welcome, Dr. Canney. It's a great pleasure to have you on our podcast. Thank you very much for joining. And can you please briefly introduce yourself and let our listeners know where you're joining us from what part of the world? Thank you.
Well, thank you so much. First off, just thank you so much for inviting me to be on your podcast. Joining you today from Denver, Colorado. Sunny and beautiful here today. Yeah, I'm the Chief Scientific Officer of Carthera, as you mentioned. I've been with the company since it was founded in 2010. Spent a lot of years living in France, the beginning of the company when we were just getting formed and had a very small team. And I moved back to the US in 2015. So I've been living back in the US for about 10 years now.
And what do you see is the difference in how we approach clinical trials and the work that you do at Carthera in Europe and France compared with the United States? Do you notice any major differences?
Yeah, I don't think there's a lot of difference between the two countries and how they approach medical research. I think that they both run rigorous medical trials to prove that a new therapy works. I think the bar is similar to getting something approved. We ran our first clinical trial, our first in-human clinical trial was started in France, and we just ran that in France in 2014. That was when that first started. And then we approached the FDA around 2000. Well, we had a lot of interactions with the FDA, even early on, very early on. We interacted with the FDA, but we launched a trial in the US in 2019 where we got FDA approval to launch that trial. And they were both very similar, I think, in how we interacted with them. The FDA has been very fair, very open to discussions, and we've had a great experience with them.
Yes, thank you. And now let's tell our listeners, what are you doing at Carthera? What are those medical trials, medical research that you are conducting?
Sure, yeah. So what we're working on is to use ultrasound in a different way. So we typically think about ultrasound, everyone thinks about imaging babies with ultrasound. This is the first thing I think comes to mind. When you say the word ultrasound, they think babies. This is our main interaction with it, whether I think we've had a child or whether we've seen it on TV. But it's pretty exciting what ultrasound can do. And there's been a lot of research in ultrasound to take it beyond imaging. And so it's been mainly an imaging modality for a long time, for decades and decades. And now been a lot of research over, let's say, the past 20 years to say, maybe we can use ultrasound in new ways as a therapeutic modality. I shouldn't say just 20 years, actually. This goes back even way farther than that. There were some very early innovators in the field who were a little bit beyond their time that tried to use ultrasound as a therapeutic modality, but maybe didn't have all the imaging guidance and other tools that you need to make it a successful therapy. But now I think we're really at the forefront where ultrasound is being used as a therapeutic modality in a lot of different ways. It's being used to replace surgery, and there's companies developing ways of using it to heat and ablate tissue. Also, companies using it to mechanically destroy tissue. And this is kind of as a replacement for surgery. We're using it a little bit differently, which is as a drug delivery tool. We use ultrasound in combination with a microbubble to actually disrupt the blood-brain barrier, which is a big barrier for getting drugs into the brain. And it's something that's been explored for a long time. And now we're kind of on the cusp of showing that we can safely disrupt the blood-brain barrier with ultrasound and improve really the delivery of drugs to the brain.
You so much. And for those who are just getting familiar with the topic, can you explain why do we need to disrupt this blood-brain barrier and what is it in general? Now, why do we have this barrier? Can you just give us a little bit detour so our listeners would understand this topic better?
Yeah, so the blood-brain barrier is really a protective layer you can think of in the blood vessels in the brain that prevents things from crossing it. And it's there for a reason. I think the brain is a sensitive organ. We don't want to necessarily let toxins get into the brain very easily. And so it was probably evolved over time to protect the brain from those things. But because of it, we can't really get drugs into the brain. And so we could start off like if you think of another organ, you may give a drug through the bloodstream and imagine that the drug crosses over into the tissue that the blood is perfusing and gets almost an equal concentration between what's in the blood and what's in the tissue. If we look in the brain, most drugs, if you give it in the blood, may only get to 1% of the concentration that was in the bloodstream. And so you're getting very little amounts of that drug across, and your possibility for getting a therapeutic effect from that drug is very limited because you may just not be able to get enough drug there.
In this case, this protection works against the brain because it cannot get the treatment that we want to give it. And you are developing this intervention that will allow to open the gate in this blood-brain barrier so that we can deliver the drugs directly to the brain, to the site that we're targeting. And in what particular disorders this would be the most helpful? What is the clinical population that you are mostly targeting?
Yeah, so there's a lot of applications. I mean, you can imagine almost any disease of the brain, we may have this issue for getting a drug. So there's a lot of unmet needs because of that. And there's been a lot of issues with drug delivery, drug development for brain indications because of the blood-brain barrier. Here we're focused on glioblastoma, which is a type of primary brain tumor, originates in the brain, very deadly. There's very few treatments for glioblastoma. Typical overall survival for patients once you get diagnosed is a year and a half. And it hits people in their 30s, 40s, 50s, 60s. So it can affect people pretty early in life. It's not just kind of a late stage disease that happens when you get older. And I think that's why I always say it's a disease where it's not necessarily super prevalent, but at the same time, everyone seems to know someone who's been affected by it. But just getting back to your question, as I talk about glioblastoma, you talked about all the indications, and I think we can think about bigger indications. There's been some early work. We did some work in Alzheimer's, for example. This is a disease that affects many more people, but also limitation of getting drugs to their target, Parkinson's disease, ALS. So this is not just brain tumors that I think we can potentially treat with this technology.
Yes, and I'm sure that there were already attempts to disrupt that blood-brain barrier before. Can you give us this a little bit historical detour of what's already been done in the area so that we can then understand more what you are providing to solve this problem?
Yes, there's been a lot of development in the field, as you mentioned. One of the areas was using something called mannitol, which can basically create a temporary disruption. It's like a drug that can create a temporary disruption of the blood-brain barrier. Came with a lot of, I think, side effects that limited its kind of widespread adoption, and maybe also had a very limited window for giving drugs. There's also been attempts at directly kind of overcoming the blood-brain barrier by just directly injecting drugs. And so you can think of sticking a catheter or a needle or whatever you want to think of directly into the brain tissue, and let's just put the drug right in there. There's been a lot of work around that. And this is a field called convection enhanced delivery, is typically what it's referred to as CED, still being explored in active research studies because of the blood-brain barrier. It has some applications, but hasn't really had a lot of widespread clinical adoption. Some of the limitations, I think, around some of these other technologies is that can be difficult to repeatedly perform them. So if you want to give a patient a drug every month, every week, whatever it is, some of these procedures are invasive. You can't leave a catheter necessarily in the brain for months at a time without having a risk of infection or something. So the technology that we've come up with that's been developed around this, I think, has a lot of advantages over some of these other things in that we think it's safer so far in the patients we've treated. We think we can repeatedly do it, so we can give drugs very regularly to patients. And at the same time as we give the drug, we can disrupt the blood-brain barrier and have this gate open for drugs to get into the brain it's very temporary, so the human body starts repairing it pretty quickly. So far, we don't see a lot of toxicity from doing it because we think it's repairing itself within a few hours. So potentially a lot of advantages, I think, over what's been done. Mentioning other technologies, there's also companies developing drugs that can cross the blood-brain barrier. And that's also been a field that's been around for a long time. It comes with other issues, I think. You can imagine doing drug development for a new compound is typically, we think of that as a 20-year process or something. If you want to start from a new chemical entity and then develop it into a marketed drug. And here, our technology can already just be adapted with any drug that already exists. So you don't actually need to re-engineer the drug. You can just use it with our system.
Yes, it sounds like a very innovative approach. And maybe you can mention things that nobody else is doing and what really sets apart your approach from everything else that's been done so far.
Yeah. Just to give some perspectives, this field that we're talking about, which is ultrasound to disrupt the blood-brain barrier, ultrasound for this therapeutic kind of modality of drug delivery, it's been around for a while. And so the first paper was published by this very brilliant scientist, Claire Bohannon, in 2001. So they did the first work. They showed maybe it's safe. They did some animal testing. And then we go from 2001. So basically in 2014, we treated, I think, the first patient with this approach. So it was 14 years to go from this first proof of concept to treating the first patient. And one of the big issues that we had to overcome was the skull bone. And so ultrasound doesn't really want to cross the bone, which is a kind of just a barrier for ultrasound. There are some ways to get across the skull bone. There's a lot of companies developing very innovative solutions to get ultrasound across the bone and do that safely. And there was a lot of push from scientists and engineers in the field to develop these approaches, and they're in clinical use now. There's some really amazing things going on in that space. But you have to buy a very large $1 million system. I don't know what the cost is exactly, but they're very large systems for hospitals to buy, complicated to use. It may take 4 hours of hospital surgeon time to do this repeatedly. And so our approach uses an ultrasound implant. And so the founder of the company, Alexandre Carpentier, is the founder of the company. He's a neurosurgeon in Paris. And he showed up at an ultrasound conference, and I think he saw these approaches going on, and he said, I'm a neurosurgeon. You guys have all these complicated approaches. I think I'm just going to put the ultrasound transducers basically in contact with the brain. And this way I can overcome the skull bone. I know that for the tumor patients that we're treating, most of them have surgery anyway, so why not use that surgical opportunity to just put this device in? We can safely deliver ultrasound that way. We can do it in a way that is very fast. We don't use a lot of resources of the hospital. And this is what founded the company, some of these ideas from him. And subsequently, we've shown that this approach is feasible, easy for the clinicians to adopt and to use regularly.
Yes, thank you for this information. And initially, I think sometimes we might have a misconception about the blood-brain barrier, that it's a barrier that if we, let's say, disrupt it, we disrupt the whole barrier across the whole brain. But actually, it's not the case, yes? We are disrupting it exactly at that space which we're targeting with the device. Everything else really doesn't get disrupted. Do I understand the process correctly?
Yes, you understand it perfectly. Yeah. So with our device, really how you place it is going to determine what you're opening. And so if we have a device that's about 6 by 6 centimeters, we open a volume that's about 6 by 6 by 7 centimeters. And so, like you said, we're not disrupting the entire brain necessarily, although there's some data that shows maybe that could be possible. Maybe that is safe. I think there's still some debate in the community. Could we disrupt the whole brain? But right now, we're trying to really disrupt the region that's left around the tumor where we know there's infiltrative tumor cells for these patients. And we're trying to kill those cells so that the tumor doesn't recur.
Yes, and I just got curious thinking about that, because when we do tumor removal, you insert usually the device at the place where we entered the brain, the place where we open the skull, remove the skull bone, and then you would place the device there. Is this always the area that we need to target, or there are some cases where a surgeon goes from the certain entry point that may be a little bit further away from the area that we need to target? The question is, are we always on the target if we're just following that initial entry point?
Yeah, so when a surgeon targets it, you're exactly right. It doesn't necessarily mean that the bone window they would kind of open up to do their surgery necessarily perfectly lines up with where they want to put our device.
Yeah.
In some cases, they move the device, they create the bone window a little bit off from where they would normally, or
Mm-hmm.
they may enlarge it a little bit and then place our device.
Mm-hmm.
For the neurosurgeons, this isn't a big issue. It's pretty fast for them, and they're used to doing these kinds of things.
Yes, very interesting. Now, how long does this disruption work? What is that window of opportunity for us to deliver a drug in this case?
Yeah, so we actually had the opportunity to measure it in kind of a unique way. Ran a phase 2 clinical trial in patients, and something interesting happened in that when you run a clinical trial, things don't always work out exactly as you want them to. And so we had patients who had this technology. They turned on our device, they disrupted the blood brain barrier, and we said, go to the MRI, get a scan, and let's see if we can see. You can use contrast agent to see if the blood brain barrier was actually disrupted. So verify that the device worked. And we wanted to do this pretty quickly in the clinical trial. So we said, go and do this and then go to the MRI. Well, it turns out at hospitals, the MRI is not always available when you want it to be available. And so
Yeah.
this can pose a big issue when you're running a clinical trial. If you say, hey, I want you in the MRI in 10 minutes,
Yeah.
it's not always available. And so you come up with these issues. In this case, it was kind of interesting because we wanted patients to be in this MRI, get the scan within an hour. Some hospitals, patients went out to 2 hours before they could get in the MRI. But the interesting thing about it was all this variability and when we could get the patient to get the scan, we kind of mined the data. We have some great engineers on our team, and we mined this data and looked at it and said, wow, we can actually calculate how long it takes for the blood-brain barrier to close from this data, because it took some patients went in the MRI in 10 minutes, some patients went in 2 hours. We said, let's use all this data and let's look at it. And so what we found is that the blood-brain barrier closes pretty quickly, and this is what we measured. It's on the order of an hour, just over an hour. It's really the half-life. It's closing very quickly after this procedure.
So that window of disruption is about an hour for us to deliver the drug. Mm-hmm, mm-hmm. Can you describe a little bit the device? How does it look like? And just basic characteristics of it.
Yep.
Oh, yeah, here it is. Wonderful. Mm-hmm. Yes, can you tell more about it? Yes. Hey.
Yeah, what I have here on my desk right now is this isn't actually the device, but we made it,
Mm-hmm.
3D printed copies of the device. And so not exactly the same color and everything, but
Mm-hmm.
it shows you the form factor for the device. And so what we have here is we have 9 ultrasound transducers. These are each 1 megahertz ultrasound transducers.
Mm-hmm.
And what we do is we have these 9 ultrasound transducers. So this is the side that's actually in contact with the brain, or we're on the surface of the dura mater in this case.
Mm-hmm.
And then on the other side, we have these transducers that are mounted on a titanium mesh. And so this titanium mesh is what a neurosurgeon would typically use if there was an issue with the bone, or a patient has an accident, or to replace the bone with something. A lot of times they'll use these mesh to actually replace the bone. So neurosurgeons are very familiar with this kind of material for neurosurgery. And on the top of the mesh, there's this port. And so we have this small port that's just protruding, and you can see from the side, it's just protruding a little bit. And so what happens with that protruding port is that once you place it, you can feel that port through the skin. And so hospitals are used to having these kinds of ports. They'll oftentimes use them to inject chemotherapy or other things into patients. And so it was designed so that once the device is actually implanted, you can actually feel this port through the skin. You can plug in a needle that actually powers the device. And so we have a single-use needle that plugs in through the skin into the device, because there's no power supply on the device. And that's for a very specific reason, is that we want the whole device to be MR compatible. And so everything in here is MR compatible. There's very little artifacts. And so for these brain tumor patients, they typically have scans at a regular frequency, and you want to be able to see, is their tumor recurring? And so if you had a large artifact, you'd interfere with that.
Can you explain a little bit more? How do you power the device, or how that needle can do it? You charge the device beforehand?
So the needle plugs into the device each time you want to activate it, and then it plugs into a touchscreen computer system, power supply that we've developed that kind of can guide the team through the treatment. There's no battery. There's no battery either in the system. And so the needle directly powers the device. So when you plug it in, the computer system checks that you're actually connected to the device. It checks that everything works on the device. And then it, in this case, sends a 1 megahertz signal to the device that then is diverted to these ultrasound transducers. We activate them sequentially so we can avoid any issues with heating, for example. We try to minimize the amount of energy that's deposited all at once. And so they're all activated sequentially. These 9 emitters gets all the energy to the system to do that.
That's amazing technology. And I know that you are also using bubbles. What are the bubbles and why do we need those? What do we need to pair ultrasound with the bubbles?
Yeah, so we use bubbles, as you mentioned. So these are oftentimes referred to as ultrasound contrast agents, is what we think of them as. They've been used in imaging for a long time.
Mm-hmm.
And what they can allow you to see is blood flow into an organ, for example, because air bubbles will reflect ultrasound. And so
Mm-hmm.
that's how they've typically been used. In this case, we actually
Mm-hmm.
inject the bubbles at the same time as we turn on the ultrasound.
Mm-hmm.
And these bubbles are also resonant at the frequencies that we're using. And so if we apply 1 megahertz signal, we're probably getting, bubbles that are on the order of 2, 3, 4 microns. We're driving basically. So they're oscillating in response to that ultrasound. And actually that oscillation within the vessels is actually coming in contact with that vessel wall of the blood-brain barrier, and it's actually causing the disruption. So it's this interaction between the ultrasound energy that we're sending and the microbubble that's creating a mechanical stress on the wall. I think that's disrupting the blood-brain barrier.
That's amazing. For how long do patients have this device implanted?
Yeah. So the patients get the device implanted at the surgery, as we mentioned.
Mm-hmm.
They'll get it activated at each cycle of chemotherapy.
Mm-hmm.
They can get it removed at the end if it isn't working, or they can keep it in. That's really up to the patient.
Mm-hmm.
We've had patients who haven't had the device removed. It hasn't bothered them, so they've just kept it in because they don't necessarily want another surgery. And other patients who have gone in for another resection procedure or something, if their tumor regrows and the device has been removed at that point.
Mm-hmm. What is the longest time they can keep this device implanted? Is it like the whole life they don't need to remove if they don't want to? Doesn't it cause any inflammation, or what is the safety of keeping this device for a long period of time?
Yeah, so that's data I think we're acquiring right now. So we've had patients who have it a couple of years out. They seem to be fine, not have any issues with it. But I think that's something we're continuing to evaluate in our clinical trials and explore.
And what did you notice after conducting your clinical trials? What is the effectiveness of this method? How does it affect the treatment compared to the standard usual procedures that are being used?
Yes, we're really lucky because we get to work with a really amazing team at Northwestern universities. We met some of them originally at ASCO, which is a big oncology conference. And Dr. Roger Stoop is a pioneer in treatments for glioblastoma. So we met him years and years ago, and he was very interested in our technology. We've been still working with him on our trial. And we also met another neurosurgeon named Dr. Adam SONOBIRD at Northwestern. And just to get to your question, he's the PI right now in our phase 3 trial, and we've been running a lot of clinical trials with him. But he's also not just a physician, but he's also, I would say, a scientist. He's got a lab and he's curious and he wants to learn as much as he can. So he does research and really amazing things coming out with him. But as part of the clinical trials, he actually wanted to say, I want to directly measure how much drug we're getting into the brain. So we had this data in, you do animal experiments, you measure it. We knew we've seen a 5-fold increase in animal experiments we've tested when we've disrupted the blood-brain barrier. Dr. Sonobin said, let's see if this really holds up in humans, because humans can be very different. Know that things you do in preclinical models don't always translate to humans. And so he said, let's see if we can actually measure the drug uptake in the human brain. And so he took patients who are going on some of our clinical trials. We disrupted the blood-brain barrier before the patient actually had a tumor resection. And so you can basically place the device, you can imagine, on the brain. He turns on the device, disrupts the blood-brain barrier, and then gave a chemotherapy in the OR. Here you can dose it a little bit lower dose because the patient has to recover from the surgery, but you can give a low dose chemotherapy and then do biopsies, and you can actually measure directly what is the drug concentration in regions I've targeted with ultrasound and in regions I didn't target with ultrasound. And some fascinating experiments he did, because we also got to use this tracer. He uses fluorescein, so you can use a fluorescent tracer to image what you disrupted. And so really incredible images. We published these a few years ago in Lancet Oncology article that he wrote. But you can actually see firsthand, with your own eyes, I've disrupted the blood-brain barrier, and it's very dramatic. It's amazing. I was there for some of the first experiments in the OR with him. And then when you go into biopsy, you take out pieces of the brain you were going to remove anyways during the resection procedure, and you actually dose the drug and you say, is there more drug, in regions I targeted with ultrasound than regions I didn't target? And what he found, which was pretty amazing, because like I said, the animal experiments don't always hold up directly in people.
Mm-hmm.
He saw almost a six-fold increase in drug levels for this drug carboplatin that we're using in the human brain. So we tested two different drugs, a carboplatin and another drug, which is a paclitaxel,
Mm-hmm. Mm-hmm.
and saw in both cases we had pretty significant increases in the drug concentration
Mm-hmm.
from disrupting the blood-brain barrier.
What are the challenges that you still see you need to overcome in this process?
Yeah, so there's a lot of challenges. I think the first challenge that we have is to show that our intervention is better than what exists right now. Patients with glioblastoma that we're treating, we're taking patients right now on our phase 3 trial who had their first recurrence of glioblastoma. So they've already had a first treatment with a chemotherapy, radiation, surgery. They get a recurrence of their tumor. It's at that point that they come on our trial. They get randomized to either getting our device plus carboplatin chemotherapy, or they get randomized to getting surgery plus the standard of care, which is one of a couple other drugs that we know at this point very ineffective. I think the response rate of some of these other drugs is less than 10% of patients have a response to them. But still, this is the bar for getting an approval for showing that something works. And so we're comparing our treatment against the standard of care and hopefully going to show that it's better for these patients than what they would normally get.
Would like now to bring our listeners back in time into your past and find out how did you get to this place where now you are conducting these amazing clinical trials? How did your interest in neuroscience in general develop?
Yeah, I mean, how far back do you want to go? We can discuss.
As far as possible, because that's the story. I think it's so inspirational and also interesting to learn how those interests develop and how does the career develop and people become who they are. What influences those decisions and career paths?
Yeah. Okay. So I will say I don't have a family of neuroscientists or anything, not any of that, but I did have a lot of interest in math and science, I think, from a very early age. And I ended up going to Boston University for my undergrad. I think I knew I wanted to go into engineering.
Mm-hmm.
I went and studied mechanical engineering as an undergrad at Boston University. And fortuitously, but I also wanted to do research. And so I think in my senior year, I went to one of the faculty members and I said, hey, I want to do research. Can I come and work in your lab? And many of the professors at that time were working in acoustics and ultrasound research, really.
Mm-hmm.
They took me into their lab. They said, yeah, here's a project. Go work on it. I worked on it a little bit and got to see what it was like to be a grad student, got to hang out with some of the grad students that were always really awesome. And they put up with me hanging around with them in the lab, I think, which was great. After that, it led me to go to graduate school.
Mm-hmm.
Originally, I was thinking, well, maybe I'll just go do a master's. I knew I wanted to do a little bit more. Then you kind of start looking at grad school programs and you say, well, I'll just go do a PhD and see how that goes. And so I ended up at the University of Washington in Seattle, and they have really great center for, it's called the Center for Industrial and Medical Ultrasound.
Mm-hmm.
And there was a great researcher named Larry Crum who founded this lab of acoustics ultrasound research, I would say,
Mm-hmm.
and ended up doing my PhD there and learning all about therapeutic ultrasound and had a great experience. So that's not quite to the neuro part, I think is what you asked about. But this is how I got wrapped up in the world of ultrasound. I think it was pretty exciting to go there and say, wow, all these new things you can do with ultrasound, especially for me, I think in the medical world, it was like the possibilities are endless. And it's pretty amazing to have been in the field for a long time because we just keep exploring new things. Like now, I think neuromodulation is a huge field with ultrasound that has just taken off. A lot of new companies being founded, a lot of incredible research in that area. But I went and did my PhD there. And then at the end of it, I actually knew some people in France who ran a lab. And so one of the researchers in Lyon at this lab called INSERM, they have a network of really great government-funded research labs that have all kinds of topics. But there's one in Lyon especially that specializes in therapeutic ultrasound. And he, sent me a message and he said, hey, I have a postdoc opportunity here. Do you want to come and live in France and check it out and do some research here? And I said, well, sure, let's go. So I ended up in France for a postdoc. And shortly after that, found out that the company, the project I was working on actually was a project initially with an academic collaboration with this lab, but then it fairly quickly turned into a startup company in France, started by, as I mentioned, this neurosurgeon, Alexandre Carpentier. And so he is a neurosurgeon, so that got me into the neuro world, and he wanted to use ultrasound for different therapeutic things in neurosurgery or drug delivery, all these neuro applications specifically.
What fascinated you in ultrasound? What potential did you see at the time when you were doing your PhD and then postdoc?
Yeah, that's a tough question to answer. What fascinated me about ultrasound? I don't know. I think it just spoke to me. I think I'm kind of a musician on the side as well. And so there's kind of this interplay with acoustics. Acoustics is a very general thing, but being able to do research and something you also have a hobby is kind of interesting, somewhat related. Mm-hmm. And what I found, at least in the ultrasound world, is there's this kind of amazing community. I think very quickly that was evident. I got to go to a lot of academic conferences, first through the Acoustical Society of America. It was a really amazing organization that's very broad in scope. I would say anything acoustics is related is encompassed in that. I went and participated in the student council and got to go to all these conferences, and I was president of the student council and ran events for students, and that was wonderful. It's a very small community, but everyone knows each other and is very supportive.
And you also mentioned that you were invited to do a postdoc in France. What qualities you need to possess, what knowledge to be invited? You did not actively apply for the position. They just approached you. Would you come? What advice would you give to our listeners to increase their chances to be invited instead of applying?
Yeah, I think that I think every lab is looking for good postdocs. They have funding. Every lab is looking for good postdocs and good PhD students for that matter. If you want to start even before that, I think I came lab as an undergrad that my PhD advisor kind of knew, and I don't know if they were even looking for PhD students, but they said, okay, we'll find funding for you. And I think that the postdoc situation is almost the same from what I've seen. I think if you are very interested and enthusiastic about a topic and want to do a postdoc in a lab, I think you can email most of the PIs from these labs and send them your resume and say, hey, I want to come do a postdoc. And you may do that a year before you finish your PhD, so you give them a heads up. And many labs will have funding or already looking for postdocs, or maybe they say, hey, you need to come with your own money, but here's some grant opportunities you can get for funding a postdoc. So I actually did have some money from the Acoustical Society of America I mentioned. They had a fellowship, postdoctoral fellowship that applied for and got, and that helped, fund my postdoc, but was kind of allowed me to do whatever I wanted. So that was pretty awesome too.
Probably you were active in the field, as I understand, because you said you were a president of a student council. You were already positioning yourself as being active, as being passionate about the field.
Yeah, and I think I also benefited from, I had a lab that was well funded. I had a lab where our advisors really, they were like, go to as many conferences as you want. I guess I realize how atypical that is, because now I talk to PhD students that say, well, I have to pick one conference a year and that's all I can do. And I think my advisors were always like, wow, go to these conferences. I would maybe go to 2, 3, 4 a year sometimes. And it was like great way to travel, great way to meet different people in the field. So it was always, just an awesome experience. And like I said, I have to thank them for allowing me to do so much of that, because it's not that typical for most PhD students, but it's a great way to see what's going on, meet people.
Yeah. And you took an advantage of these opportunities, which was amazing. Communicating, collaborating, building networks at those conferences, in addition to learning about new things that are happening. Yeah. And I also noticed one very interesting thing here that I think will be relevant to many people who are listening to us, because we have people coming into the field of neurotech from engineering. And their question is, how much of neuroscience should I know to work on the projects in neurotech? You were in a similar situation in a way. You were working with ultrasound more from engineering perspective, and then you're meeting a neurosurgeon, and here the work related to neuroscience begins. So how did you approach this? How did you learn? How much did you need to learn to get into this work and work successfully?
Yeah, I think you touch on something that's maybe one of the coolest parts of the field. I guess anything you do, but is the ability to work with people where you really have great experience. But wouldn't say I knew much neuroscience, and I'm still consider myself a novice in most ways. I'm learning, I'm trying to learn as fast as I can, and you're always reading papers and trying to learn. But you try to work with people who complement their specialties. And so we get to work with neurosurgeons and we respect them, and we have a way of communicating and learning from them and asking questions and getting their expertise. And they don't know anything about ultrasound, maybe. And so the two together is strong. And you don't necessarily need to be the expert in your domain, but you need to just be able to discuss and learn from people and always be learning and open to that. And I think that's probably the most important part.
When you were already finishing your PhD, or maybe during your PhD, and probably even earlier, did you already think about what career path do you want to take? Will it be more academic, becoming a PI and having your own lab, or rather taking a path in industry, which you ended up doing? Yes. How was it developing for you?
Yeah, I think I never really saw myself as a PI or a professor. I don't know why. That was never something that excited or interested me. So was to become just a pure researcher. I love research, and I think somehow I found that balance because I get to collaborate with amazing researchers and kind of benefit from learning from them, but I don't have to apply for NIH grants and worry about funding and all these things that are ongoing. I mean, it's very stressful, I think, right now to be a researcher in academia in the US, at least, with everything going on. I've always taken a little bit more of a circuitous route, I think. I always wanted to live in a foreign country, and I used the postdoc as an opportunity to do that. I wanted to be fluent in another language, which I wasn't, never really learned. In the US, I think we don't value being proficient in another language. We take a few language classes, but we never really become proficient. And so I ended up saying, well, I'm going to do that now as a postdoc, and became fluent in French basically by living in France, living in a foreign country. And that was fantastic. I think as far as career trajectory, I think I've been late in kind of like having a well-defined career trajectory. I don't think I necessarily had a path that says, well, this is where I want to get to. I think as long as you're working with interesting, inspiring people, I think that's the most important part. I don't know. Maybe people function differently and say, I want to get to this next step. But I think as long as you're continuously learning, and I think a startup company is interesting, if we step back and look at a startup company, because your role is always evolving. I was started out at the lab bench doing experiments and doing animal experiments and programming and doing all these things that you do as an engineer. And you grow through each stage through learning about the regulatory steps, through working with clinicians, working with clinical affairs teams, working every year almost it changes into a new almost job and role. And I think that's the exciting part about any career, I think, is always learning new things.
Yes, that actually was my question. How did your career develop over those years? Where did you start? What was your initial position? And how did you come to the place where you are already chief scientific officer in the company?
Mhm.
How did this all work out for you?
Yeah. Yeah, it's been quite a journey. It's as I mentioned, I mean, it started out really me and another engineer at the company were the first two hires, and we worked together. He kind of managed a lot of the regulatory stuff and different suppliers and things, and I worked on testing the device and getting the first generation system ready for using in humans originally, and showing the safety in preclinical models, and then getting it ready for human clinical trials. And so we had a very small team, it was like
Mhm.
a few people, and we had some interns and we had some other people along the way, but very lean team to get the first trial off the ground. We were lucky really to leverage, like I said, this lab in France. We kind of were able to get incubated a little bit and use their facilities, resources, and have access to their researchers and everything, which was also a big help in the early stages of the company. We grew a little bit more. You as a startup, you grow each time you raise some more money, helps to, pay a few more people. But we grew a little bit more. I think around 2014 is when we raised some more money and hired a CEO. We had a kind of an acting CEO before that who helped with a lot of the legal administrative aspects. But we hired a CEO that was a little bit more 100% involved in the company at that point. Then we hired chief clinical officer and some other key positions in the company to grow further, but still stayed as a very small team. I mean, we were, I don't know, 10 to 15 people until a year and a half ago. Just very, I would say, people who were able to do a lot of different things, weren't really specific to one task. We each had our area, but in the early stages of the company, I think you want people that are at least willing to take on anything, even if they don't really know how to do it. And as you grow, you end up with people who are more specific and working in very focused areas. And so now we have quite a big team. We have 30 people now, I think, at the company. We have many more people in the US. We were just a French company focused in Lyon and Paris. Now we have 5 people in the US. And so that's been a big transition, is going from a French-speaking company to an English-speaking company. We've had to impose English on the French team, which has been quite a change, but going smoothly, and it's a good team. It's a great team.
That's a nice team already. And what were the biggest challenges in this path that you went from the beginning until where you are now? And how were you able to solve those challenges?
The biggest challenges, I mean, there's so many challenges. It depends on what specific area you want to, as keeping the company funded. There's the regulatory aspects. There's the day-to-day, I think, of staying excited about the technology as you have new technology has quite a long development time. And so, continuing to stay enthusiastic, I think, is always a challenge. But you always get these kind of nuggets of things, like you'll see a patient do really well, and that will give you some new encouragement, I think. But yeah, I think, there's been a lot of challenges. I think we've been really lucky to hire some really great people that have made our development possible. And so I think hiring the right people is one of the biggest challenges, because if you hire the wrong people, you can get stuck, you can get frustrated, you can have people leave. But we've been lucky to hire some great people along the way to build out the company, and that's been great.
And that's a great topic, because, of course, people who are also listening to us, they are applying for positions. They want to get into the companies like yours. Can you tell a little bit more? What are you looking for when you are searching for a candidate for a position? What are the key things that you think are important in your potential candidates?
Yeah, so this has been a big focus. I've hired a lot in the past year. I've built out one role. I'll speak specifically. We have clinical application specialists is the role. So we have people on site to make sure that someone implants their device or does a treatment, that they do it properly. And this was a role that was kind of just created in our company in the past years to support our clinical trial, one of the many roles that you need, I think, to support a very large trial like this. So along the way, I've gotten acquainted with hiring very intimately, because we've been interviewing a lot of candidates and seeing would they be a good fit. The most important thing is probably curiosity. I mean, you want people who are curious and people who come to an interview and ask questions and want to understand things. They don't need to understand everything, but you need to be curious, and they want to have an interest and want to learn, continuously learn, even on their own. So you don't want to have to tell someone, hey, go and read this, go and do this, go and do this. But we have team members who are coming to me and be like, well, what do you think about this? Or I saw this paper. What do you think about this? And I think that kind of intellectual curiosity is probably the number one thing you want to look for. And then after that, it's people who are self-starters. In a startup company, you can't really monitor everyone and give them a checklist and say, hey, did you do this? Did you do this? Did you do this? Did you do this? You kind of need to rely on people to work on their own and complete tasks and not have to really watch over them, especially with a remote team. I mean, you need to trust that they're going to get things done, even if you're not talking to them every day and following up with them. So these are a few of the things I think about, maybe general things. I think that the other thing is cultural. There's always, I think every company has their own culture a little bit. We have kind of a unique culture as a French origin company, I would say. And so you need to think about, is this person going to be a good cultural fit for the company as well? And I think that's important, because otherwise I think the person isn't going to be happy at the company, and the colleagues may be frustrated as well. So
yes. Where do you see the field of technologies like this is going? Or what we can see more in the near future, as right now there is such a fast technological development?
Yeah, I think if we just back up to the whole field of therapeutic ultrasound, which is maybe where we somehow started, the number of applications is growing. There's an organization called the FUS Foundation that has done a great job at getting more PR and things around this whole technology field. But they've tracked the number of approvals that have happened in companies, and it's been incredible. I mean, explosion in the past decade or so in this whole field. And so we're seeing FDA approvals, we're seeing adoption of new technologies, and so it's growing rapidly. I think that for the field that we are working in, which is drug delivery in particular, we're just at the early stages. So there's no approved devices to do ultrasound-based drug delivery, at least that I know of. Maybe I'm going to offend someone if I miss something, but there's a lot of companies out there exploring this, and I think there's a lot of potential. So we're really just at the early stages of getting this to patients. And once we can show the first signals, we're running our phase 3 clinical trial, which will hopefully show that we can have a benefit for patients. And if we do, I think that's just the start because, like I said, we're combining it with one drug, but we're also exploring through preclinical collaborations, through early stage trials. We're exploring all kinds of therapeutics. So we mentioned how the blood-brain barrier really limits what therapeutics can cross. I mean, 98% of drugs can't get across. If we can open this to many more different drugs. Future for me is to cure this disease. I think that's what I would like to say. If we can cure this disease in my lifetime, that'll be a big success.
Yes, absolutely. And this is one of the things that you mentioned that drives your passion to help people and lead to final cure of the disease at some point. Speaking about this possible future development, what options do you think this development opens in terms of careers? Where people in the future, coming from engineering, coming from neuroscience, can find themselves? What can they do? How they can contribute to the field?
Yeah, I mean, I think the number of careers is just expanding massively. I think as we get adoption, as we get reimbursement for these technologies, as we see companies be successful, that only drives more investment in the field. And that means more companies are going to get created, more startups get formed. That all means more job opportunities, I think, in the space. It's kind of a reinforcing cycle as we grow. But there's a huge number of companies being created, not just in this drug delivery space, but as I mentioned, for therapeutic, I would say replacing surgery. That's a big area where companies are working in. And for neuromodulation, we're seeing a lot of companies get created in this space. And we'll see. I don't know. There could be something else, something new that comes on the horizon that generates even more excitement, but it just keeps growing.
Yes, and as you just said, that things come on the horizon. There are more and more technologies that are being developed. Is there anything that inspires you, any new developments that maybe make you even more passionate and curious about the field?
Yeah, I mean, I think for the specific area we're working in, I think that the thing that inspires me the most is maybe personalized medicine is can we figure out what patients respond to a drug? Can we give patients the best drug possible to ensure that they're going to have a response? That's something we're working on. We know that most oncology drugs don't work in every patient. We know that there's patients who don't respond. And so if we can pair some of these developments that are happening in personalized medicine with our technology to deliver drugs, that's where I think we can get very big effects potentially.
And we know that we have an explosion of AI currently, and it already influences many areas of neurotechnologies. People are developing speech neuroprosthesis because we have those huge natural language models. How do you see the impact of AI on the field that you are working in?
Yeah, I've been thinking about this a little bit recently. The ability maybe to mine data. I mean, we have a lot of data. We always search for different correlations in the data. Maybe we can use AI tools to look at it differently. That's probably the main way, I think. Eventually, I think, I don't know, can we use some of these tools and all the data available to run more efficient clinical trials? That's still an open question. I think something that hopefully will come about. But right now, we're still running clinical trials where we always need to have a group that we're comparing it to, and that's getting a standard of care treatment. Maybe in the future, we can use some of these tools to definitively say we're having an effect, or I don't know, to mine the data and show an effect with less patients being treated. I mean, this would be phenomenal.
Yes, so definitely there are opportunities in front of us. And I would like to see the progression that you need to have in order to finally get your intervention approved and start working in the hospitals. Maybe you can just give us a brief overview of how this process works. What do you need to go through to finally get from the place where you just started to the place where your intervention will be utilized in the hospitals worldwide?
Yeah, so trial we're running, if we just step back further than that, and the trial we're running is a phase 3 clinical trial, randomized clinical trial. Like I said, so we're using our device against standard of care. The end goal is really to show if we're better than standard of care. And so the endpoint that we're using is the patients live longer. Unfortunately, like I mentioned, the patients with recurrent glioblastoma that come on our trial, most of them live about a year. So very short life once your tumor recurs. And so if we can show that we can prolong the lifespan of these patients who get our intervention over the standard of care, that's something we can then show to the regulators, obviously, and say, hey, we can benefit patients. We've shown it, and that can lead to regulatory approval. So that's what we're hoping. Our trial is quite large. Like I said, between the US and Europe, we have 40 centers. We're recruiting over 500 patients to show this effect, to show that we can actually have a significant impact on patients. Once we do that, though, It's pretty easy, actually, for a disease like this, I think, because there's such a high unmet need for this disease, because there haven't really been many innovations for the treatment of these patients. Once we can show that we're better than the standard of care, the FDA, I think, will approve it fairly quickly. I think the European regulatory authorities will be about the same, and then we'll be able to bring it to more patients. Obviously, once you get approval, that's just the first step. Then you need to work on getting it reimbursed by insurers and actually rolling it out to hospitals. But right now, we're really focused on running this trial and showing that we can have a therapeutic effect and getting regulatory approval from the FDA and the European authorities.
How long do you think it will take if everything goes by the plan and you get all the approvals and testing on time? How long will it take for people to actually see your device, your approach in clinical settings?
Yeah, it's hard to say. I think we're hoping within a few years. We need to finish recruitment into our trial, which we're hoping to finish next year. And then we need to wait and see enough outcomes of patients before we can really see if it had a therapeutic effect.
Mm-hmm.
So once that happens, we show that we had an effect, then approval process can go quite fast. I think in 6 to 12 months, you can potentially get approval. So hopefully in the next few years, I think we've designed our trial as well. So we have some earlier looks at the data. You always try to see, do I have a signal? Do I have something going on earlier? And we have some interim analyses built into our trial so we can get some of these earlier looks at the data. And if they look good, then maybe we'll get approval even sooner.
That is amazing. And of course, I wish you to get it as soon as possible to start helping people on a large scale. And our podcast is called Neurocareers, Doing the Impossible. Is there anything in your career which it's been fascinating? To me, it is absolutely fascinating. So was there anything that you thought as being impossible that you actually proved to be possible? And how did you do that?
Oh, you're putting me on the line here. Proving the impossible. There was a lot of skepticism early on about using an implantable device. And so not among neurosurgeons, but among engineers and researchers had a lot of skepticism, I think. I'm not going to say it was impossible, but I think that coming with the first safety data showing, hey, this is possible. We're treating patients. They're doing fine. They're not having a lot of adverse events. We're getting disruption. It's relatively safe. This was a little bit of an impossible feat, maybe. And I think even when we treated the first patient, it was very nerve-wracking because we thought we did everything we needed to show it was safe, but you still never know. I mean, you can imagine developing a new drug and dosing that first patient and crossing your fingers and saying, wow, we don't know what's going to happen. So I think overcoming that is definitely a huge thing.
Yes, it reminded me our first epilepsy surgery. Now we opened here a new center 13 years ago, comprehensive epilepsy surgery program. And we had our first patient and everybody was there. So many people just watching, the whole team. And of course, I was there. And I remember this feeling of the first patient. How will it go? How will the surgery go? Now we have all this equipment, everything, new stuff. How will it work? It worked well. There was one moment when we all paused, but everything went well. I understand your feeling about the first one very well, but I'm glad that it also went well and that you continued. And for our listeners, what would be your advice if they want to follow your footsteps and follow a similar career trajectory?
Yeah, I mean, I think I would say find out what interests you. I would say reach out to labs that are doing work that you're interested in, labs or companies. I think either one. Just directly contacting people is always an interesting way to go. I think if you're going to do a postdoc, for example, and you reach out to a lab and say, hey, I'm graduating and I want to do a postdoc in your lab, I don't know many PIs that would say no or be negative. I think a lot of PIs are going to find the money or find a way for you to come because they're saying, wow, this person has initiative. They've contacted me. They really want to come. That's probably the first step is figure out where you want to go and reach out to people and have a conversation with them.
And like you already mentioned that, be self-motivated. Yes, be curious about the topic, which is very important, helps a lot. And also based on what you already told us about yourself, be active in the field. Talk to people, network, go to the events, present your data, and this definitely will help you tremendously. Thank you very much, Dr. Canney. And as we're nearing the end of our podcast, can you also share the information about where our listeners can learn more about your company, about the clinical trials you are running, and possibly contacting you if maybe there is some mutual interest or a possible collaborative opportunities?
Sure, yeah. So we have a few different websites. More information on our phase 3 trial in recurrent glioblastoma is available on SONOBIRD.eu. SONOBIRD is the acronym, the short name we came up with for the trial. SONOBIRD.eu has more information on the clinical trial, more information on the companies on our website. That's Carthera.eu. But probably the best way, if you want to stay informed about news and different events, is probably to go on LinkedIn also and give the company a follow, because we are always posting stories and different things going on in development. So that's a great way to get that in your feed and be updated on what's happening.
Yes, absolutely. I also recommend LinkedIn as a networking platform and keeping informed and staying in touch. Thank you. And we will add all this information into our podcast notes so our listeners can find it. We went through many questions. Thank you very much for so patiently answering all of them. I appreciate it. Is there anything maybe I didn't ask, but you still want to mention before we end our podcast today?
No, I think it was pretty comprehensive. I think you asked all the questions, so I can't think of much more.
I've been curious. So Thank you. Thank you very much, Dr. Canney. It's been a true pleasure speaking to you. I wish you all the best, and I'm sure our listeners are joining me on this wish for you to succeed in your clinical trials and help so many people with this really devastating condition. Thank you.
Yeah, thank you so much for having me on.
Dear Neurocareers podcast listeners, thank you for joining me and my incredible guests on this exciting journey into careers in neuroscience and neurotechnologies. I hope you've been inspired by the stories of those turning groundbreaking ideas into impactful realities. If you are looking for more guidance on advancing your neurocareer, book a free consultation with me, Dr. K, your podcast host and Neurocareers coach at the Institute of Neuroapproaches, the only career service dedicated specifically to the needs of professionals in neurotech. Let's take the next step toward your neurocareer success together. In addition to free consultations, the Institute of Neuroapproaches offers a variety of services designed to help you thrive in neurotech. First, professional development planning. We help you create a tailored career development plan, identify knowledge gaps, and equip you with the tools needed to achieve your goals in neuroscience and neurotechnologies. Second, resume and cover letter review. Get expert feedback on how to craft documents that are specifically designed for job opportunities in the neurotech field. Third, interview preparation. Sharpen your interview skills through mock interviews and targeted feedback, including soft skills, hard skills in neurotech, BCIs, e.g., MEG, live coding, and presentation preparation. 4. Networking and job search strategy. Learn how to effectively network and uncover job opportunities while standing out in the competitive neurotech job market. 5. Negotiation and salary advice. Receive expert guidance on negotiating salaries and benefits and determining a fair salary range for your desired position. All this and more can be found at www.neuroapproaches.org. Again, www.neuroapproaches.org. So, what are you waiting for? Let's navigate the path to success in the world of neurocareers together and turn the impossible into possible. We're looking forward to working with you.